Hi there, everyone, and thanks for joining me today. Today, we're going to discuss video 3.2.1.1, which is going to cover helper CD4 plus and T cell activation for the scope of Bio 265. To kind of get started with it, we kind of want to just kind of go over some basic understanding of where the T cells and other lymphoid cells come from. So T cells and B cells alike both originate in the bone marrow, where they differentiate from progenitor lymphoid cells. Once they are born, unlike a B cell, the T cell will actually travel to the thymus and be matured there. It'll actually go through a step procedure where it is positively and negatively selected in order to be able to function well. The first thing that needs to happen is that the T cell needs to be able to develop a functional T cell receptor. That's this purple thing right here up at the very top. Once it's able to do that, it will actually be tested against antigens to make sure that it actually functions properly and will be able to bind properly. That's how it is positively selected. For negative selection, the T cell then needs to make sure that it is not able to recognize our own proteins, and so that it can't be and cause any autoimmunity in our bodies. If a T cell is able to functionally do that, it will actually develop two different classes of molecules on it. Today, we're focusing on the CD4, which is going to be representative of this molecule right here on this T cell. The other one we will discuss, which is a CD8, but we will discuss that in a later video. Once the T cell has been differentiated and has been approved and matured, it will actually leave the thymus and then travel outside to secondary lymphoid organs. Just to kind of give you another view of this, this is where we've got our CD4 molecule. This is our TCR, which will bind to the antigen, and this is our helper T cell. As I previously mentioned, once the T cell is matured in the thymus, it'll actually leave and go to the secondary lymphoid organs. Now, kind of transitioning, we just kind of want to discuss a certain situation that may occur. And you may have read this in the textbook as well, but imagine where you're walking along barefoot through a woody area and you accidentally step on a piece of wood. And in this piece of wood, you actually get a sliver that goes right through your foot. This sliver, as it goes through your first line of defense, your epithelial layer, it actually introduces pathogens into your system. These pathogens can lead to severe infection and can cause some severe damage to your body. In a way to combat that, part of your second line of defense is we have macrophages that actually will come in and ingest this bacteria and then break it down in order to be able to present this product and different peptide antigens onto our MHC2 molecules or proteins that will then be used to activate T cells. This macrophage or antigen presenting cell will then leave the infected area and travel to the nearest lymph node, where it'll actually then be able to interact with our helper T cells. At this point, our helper T cell isn't fully activated. It is just a mature naive T cell. But once this macrophage finds an exact T cell that can actually bind to this antigen, it will bind by binding the TCR to the antigen, and then our CD4 will bind to the MHC2 molecule. This is the first step of T cell activation. The second step is the presentation of co-stimulatory molecules that interact between the dendritic cell or antigen presenting cell and the mature naive T cell. The third step of activation will be the release of cytokines from the antigen presenting cell onto the mature naive T cell, which will then differentiate it into different helper T cells, but specifically a TFH cell. Once our TFH cell is activated, it will be used to further activate other components of the immune response, such as B cells, and also cytotoxic T cells. As for right now, that pretty much covers exactly what we need to discuss for our helper T cell activation for CD4 positive T cells. Thank you guys for watching, and I hope you enjoy the next video.